Did Wednesday's lecture go okay for you guys? Any questions about anything left over from there or from Monday? Okay, no problems with the homework assignment this week? Okay, so you'll get a new homework and recitation later today. That'll be on Blackboard at the end of the day. And other than that, I guess we can get started with today's material, which is getting into more practical, kind of hands-on issues in working with protein structures. What I want to do is give a bit of brief introductory information about some of the things we can do with protein structures, and then get into the hands-on part of how do you access protein structures, how we can learn about them, what kinds of information can you get from working with structure databases and other structure-related resources. All right, so one of the things we would obviously want to ask is, what can we do with a structure database? So very often we're interested in structures literally because we want to look at them. So if you know something about structural biology and maybe you're studying an enzyme protein, it might be that you're interested in the mechanism behind that enzyme. And often simply being able to see the structure and especially see how molecules with which it interacts map into the structure can be enough to give us some good hypotheses about what the mechanism of catalytic action might be or other properties that may be useful in understanding how that protein works. I mentioned previously that sometimes we're interested in structure as a source of information about evolution. So as I've discussed briefly before, we often get information about evolution from different kinds of data sources, and different sources can give us information about different aspects and different timescales of evolution. Protein structure tends to be a relatively slowly evolving kind of biological information, much slower than amino acid sequence, which in turn is slower evolving the nucleotide sequence, so it can give us a way of understanding evolution on much longer timescales. It's not the primary kind of data we use for that, but it is available for that. And one of the big uses is something you heard a bit about on Monday. We are often interested in structures because if we can get a structure related to a protein of interest to us, we can often use that as a way of getting at least a first-pass model of the structure of the protein of particular interest. I referred to that before as homology modeling. So often we would need to access structure databases because we have some new sequence and we're looking for good templates for homology modeling to build a model we can work with for the structure of our new sequence. One other use I wanted to say a bit about, because this is a computational biology class, is that a lot of what we want to do with structures really depends on computer models and computer simulations of how these structures would behave. So I know a lot of this fits more cleanly into the second half of the class, and you'll learn some of the mathematics behind these kinds of techniques in the second half. But I do want to speak a little bit about some of the principles that go into structure modeling, and in particular into how we simulate the behavior of protein structures, which largely depends on a technique called molecular dynamics and various related sorts of techniques that go by names such as Brownian dynamics. So molecular dynamics basically comes down to simulating the physical forces that act on a protein or another molecule or a set of molecules, and allowing the molecules to behave in a computer as we believe they would behave really under those forces in an actual functioning living system. And as with any kind of computer model, that's going to be imperfect. It's an approximation, but these approximations keep getting better and better and reach the point that often we can get very good information that can be quite useful in a lot of contexts where we're working with protein structures. So I do want to give at least an intuition to you guys of how that works. So if we look at a protein model like this one, so this would be a protein structure, we might be interested in trying to figure out the forces acting on this. And the way we do that generally is by estimating energies of the structure. So you try to estimate the energy, and as we'll see shortly, if you can estimate the energy of a structure, the free energy of a particular shape of a particular protein in a particular condition, that's basically all you need to be able to get good models of the forces acting on it and then simulations of the behavior of that structure. Now, it might seem that if we take something very complicated like this with a large number of atoms, that it would be hard to do a realistic model of all of the forces acting on all of those atoms. But fortunately, this is the kind of analysis that computers can do much better than human beings. We can take this very complicated system and try to break it down into a lot of small steps, and then put all of those together automatically to come up with a good model of the energy of this configuration. So basically, we would take this whole protein and we would try to break it down and look at it one atom at a time, or one pair of atoms. So we could look at a little piece of this, so this is one side chain sitting in one amino acid of this protein, and we could focus on a single atom. And we could ask, according to our knowledge of physics, what are the forces acting on this atom? Or more precisely, what is the free energy of this particular configuration of this atom, given all of the neighboring atoms around it? The way that's commonly done is to use what's called a force field, or an energy function, which gives us an estimate by trying to accumulate over all of the different contributions to the free energy of this atom. I'm going to show you how this works for a particular relatively simple version of this called the Dryden force field. There are much more complicated ones that go into a lot more detail here, but this one conveys the basic ideas behind a generic energy function. And the way we would do this is we would look at the atom we're trying to focus on, and we would ask, what are all of the ways energy would act on this? So what are all of the contributions that would contribute to the free energy of this atom? We could say that this atom is covalently bound to a couple of other atoms, and those covalent bonds could be exerting some forces. There could be some potential energy in those bonds. A covalent bond would have an ideal length for any particular pair of atoms in any particular hybridization, and we could look up in a table what that ideal length is, compare it to the length of that bond in the actual structure at an instant in time, and based on the deviation between these, assign some potential energy to that particular covalent bond based on how stretched or compressed it is relative to its ideal length. Angles in a molecular structure would have ideal angles, bonds would have ideal angles between them. So if we knew that this was an sp3 hybridized carbon, that would tell us that two single bonds here should have a particular angle between them, and the atom would be expected to have potential energy if these two neighbors are compressed relative to that ideal angle or stretched out relative to that ideal angle. And we could accumulate a contribution for that, for all of the pairs of bonds around that angle. And likewise, there would be other contributions from the twist of this bond relative to the ideal, called inversion, which reflects whether particular atoms are more favorably sitting in the same plane as their neighboring atoms or out of the plane, as well as various what are called non-bonded forces, so van der Waals interactions with nearby atoms, electrostatics, if we have charges on our atoms, hydrogen bonding as appropriate, and so forth. And a force field would have simplified models of all of these different contributions. So we could say, for example, that a stretch force we would get by looking up in a table the ideal radii of the bonds, the lengths of the bonds based on the types of atoms we have. We would look up some other constants in the table that tell us what the coefficients should be, and get a basic model saying that we can approximate this with some simple function. Now, the exact energy that from even one of those single contributions is a very complicated thing. If you really wanted to compute that, you'd have to get into the quantum theory of what's going on. Even just two atoms, that's an incredibly demanding simulation to do. But you can do a pretty good job with simple analytical approximations, little formulas that get each of these contributions approximately right. And by doing that for all of the interactions of that one atom, and then repeating that for every atom in the protein, you can just accumulate all of these individual energy contributions and get a model of the overall free energy of that protein. Are there any questions about this at this point? Okay, so that gives us the basic capability of taking a protein in a particular shape or conformation, maybe a group of proteins, and getting an energy out of it. And as I mentioned, once you can get energies, you basically have everything you need to do simulations. So we need a little calculus and a little basic physics to do that. But one of the things we can observe is that if you can calculate energies, then you can estimate derivatives of energies by perturbing your atoms a little bit and seeing how they change the energy. The derivative of an energy with respect to distance is a force. So if you can estimate energies, then a few calls to your force field lets you estimate the forces acting on the molecule. So you can see if you hold your protein, maybe stretched it a little bit relative to its ideal shape, you can figure out what the restoring forces are implied by the model of physics you have. Once you've got forces, you can then go to your introductory physics, and you can see that Newton's second law tells us how forces relate to accelerations. Some basic calculus tells us how accelerations relate to velocities. And then we can apply some techniques that I'm not going to go into here, but that you would learn about in the second half, so 252 half of this class, to take these models of the differential equations describing how the forces act on the protein and get simulations that would let us virtually throw this protein into a model of the physics and see how it might change its shape acting under these physical forces. So any questions about any of this? Okay, so I know I'm not explaining this at a level of detail where you would be expected to be able to write up a computer program or code this up. At this point, you will be doing some models of maybe not specifically molecular dynamics, but other versions of this kind of what's called numerical integration to simulate in 03252. But for the moment, I just want you to understand at a high-level idea what these techniques are and more or less how they work, so we can understand how these techniques are used in working with protein structures. Any questions about that? All right, so this lets us do a lot of different things once we can simulate the physical forces acting on a structure. We can take a structure and we can simulate with some additional machinery fluctuations in the structure over time. So protein structures are usually not rigid entities. They have parts that may move about freely in solution, hinges that may move between a small number of different positions. They often undergo what are sometimes described as breathing motions. They kind of change periodically over time, and we can simulate those kinds of behaviors, which may be very important in understanding the function of the protein. Often we're interested in using these kinds of models to optimize the quality of a structure. So very often when we create a protein structure, whether experimentally or computationally, it's not going to be exactly an ideal structure. So you can take an X-ray structure, you can thread the backbone of your protein, kind of position all the side chains in a reconstructed electron density from an X-ray model, but usually you're not going to get everything right directly, and very often you need to throw some optimization in there. Basically do these simulations to let it relax to a lower energy state to really get a good quality structure. That comes up also in homology modeling. So a key step in getting a decent homology model is that you have to do this energy relaxation in order to basically pick up the differences between your homology template and the actual protein you're trying to model. We would often use these kinds of techniques for what are called docking models. So docking basically refers to what we get when we put multiple molecular structures together. It can be putting two proteins together in a computer simulation and asking how would these attach? Do they attach with a favorable energy? Maybe how strong is that? It could be modeling a small molecule such as a ligand we think binds to this into what we believe to be the binding site of the protein, see how it sits most favorably, and maybe get ideas about which molecules bind favorably with this protein, or how they would bind, or maybe what groups might be close to them that could be involved in enzymatic interactions. We can get energies of these interactions, and we can try various kinds of virtual experiments that might be difficult to do in the lab. So to try a virtual mutation, changing some of the amino acids, and seeing in the computer simulation if this seems to affect the function of the protein in a way that might be of interest to us. It's usually the case that anything you can do in a computer is going to be faster and cheaper than anything you can do in the laboratory. So very often it's a good idea to try these simulations, what's called in silico, so in the computer, before you actually go into the lab and do them, to at least get an idea if it's likely to work. So any questions about that? All right, so there are a lot of uses in practice. I've gone over some of that briefly. Until recently, these things were very limited by computational resources. So These are relatively costly computations, doing these repeated calculations of many different force contributions over many different atoms, trying lots of perturbations. So these are pretty computationally demanding simulations. They're not traditionally something you could do well over long time scales without a significant amount of computing power. So people who do these simulations often talk about things happening on scales of femtoseconds. A femtosecond is a very small amount of time. When we're talking about proteins, because we're thinking at a small scale, we're looking at very small structures, often what we're interested in happens on time scales much faster than we would think about in our day-to-day lives. But even in terms of protein motions, 10 to the minus 15 seconds is a very short amount of time. So traditionally, you couldn't do too much. You could look at relatively short time scale behaviors, but something like folding a protein would be way beyond what's doable. You couldn't take an unfolded protein and actually watch it fold. That's really changed dramatically in just the past few years. So the latest specialized supercomputers can now get these molecular dynamic simulations running in the range of simulating milliseconds of time, and milliseconds still may not sound like much, but that's getting into the range where we can start doing really significant things with proteins, like allowing small proteins to completely go through their folding process, looking at for a protein is long time scale dynamics, getting good samples of possible states they can take on. So this is one of these technologies where what was doable 5 years ago is radically different than what's doable today, and probably will be very different from what's doable 5 years from now. So these techniques are already in wide use for anyone who works with structures, but I think it's a technique where these are going to be seeing more and more practical use in day-to-day laboratories. So this is something I think you guys should know about. It's becoming much more useful and much more easily accessible to do really serious molecular dynamic simulations. So any questions about MD or these simulation kinds of techniques? Okay, so that basically is what I wanted to say about how we would use these things. But then I want to get into, as I mentioned, the more hands-on issues of where do we get structures? Where do structures come from? Or how can we find the structures of interest to us? So what I'm going to do is break out of these slides and go to a few different ways we can access protein structure information. So you should all recognize this. This is the gQuery website. This is one of the places that I said is often a good starting point for getting biological information. And structures are no exception to that. You can get a lot of interesting information about protein structures from here. So I'll just type in a query here. I'm going to type EGFR, and I will type a modifier and human ORGN. So this is saying I want to search for something called an EGFR. That's a symbol for an EGF receptor protein, and I'm looking specifically for human versions of that. And what we can see is that this pulls up hits in various NCBI data sets. So we've seen some of these we've seen before, like the gene data set. What I'm interested in right now is hits to proteins. So I will scroll down a little bit here. And we can see that there are some nice categories of protein-related hits. So NCBI collects some protein databases. One of them, which we'll be seeing shortly, is the Conserved database. So NCBI has a nice collection of information on protein domains, something you guys should know about after Wednesday's class. That's going to be very important in general. Here it's saying that we have no conserved domains associated with that query, but we'll see there are other ways of getting at least homologous domains, so examples of domains in the sequence. But we do have a number of hits to a database called Structure here. And we can go into that, and that will give us one way of finding structures associated with this query. You need to be a bit careful because these aren't all going to be structures for this specific protein. They're just things that match that one of these up, and we can see that this is giving us information about a particular piece of this protein, particularly the extracellular region, and we can see that there is a nice 3D structure of the extracellular region of the EGF receptor protein. So there's a lot of other associated information here in this database. I'm not going to dwell on it because structure is one of the cases where I'd actually say NCBI is not usually the best place to start. There are more standard resources, and we'll get to that a bit later, but you can get to the structured information, and often it's useful to have this link to all of these other resources, some of which we've seen, some of which we'll be seeing later in the term. So just be aware, this is one place we can find information about structures by searching on a keyword, such as the name of a protein. Any questions about that? Okay, so another way we've seen to search for information is BLAST. Often we might have the sequence of a protein, and we would want to know things about that protein based on its sequence, and structure is no exception for that. We can use sequence searching, such as through the BLAST server that we've seen before, as a way of finding information about the structures of a protein. So I'm going to click on Protein BLAST here, and I'm going to take a query sequence. So if any of you want to do this on your own, this is on Blackboard, listed as the Lecture 8 query, just a piece of amino acid sequence. I'll plug that in. And then what we can do is do a BLAST search for this sequence. Now, one thing we will want to change here is the database. So this is set to the non-redundant NR database. That's basically everything in GenPept. If you're looking for structures, it's often useful to restrict this to Protein Data Bank PDB. So the PDB, which we'll be seeing later, is the standard repository for protein structures. So if we blast against the PDB, that's blasting against a database specifically of sequences that have a known structure. So we can take that and click blast. Now what that will do is bring up some hits. And before we get to the actual BLAST results that have come up, I want to illustrate one thing you might have noticed before but that we didn't really talk about, and that is this diagram that we see here. So this is a diagram of conserved domains found in this protein. So the NCBI BLAST server is integrated with its own domain database. So I mentioned that there are various domain libraries out there that work by different methods. NCBI has its own. And typically, before you even see results of the search against the database you picked, it can very quickly run against this conserved domain database and pull up the fact that there seem to be 3 basic domain regions that can be annotated in this particular protein. So that conserved domain search is generally very fast. So NCBI does this by basically having a consensus sequence for each domain in its library. That's a relatively small database, and it can just quickly blast your query against that database and pull up these hits. And if we click on that diagram, we can get more information about conserved protein domains found in this particular protein. So we can see here that there are 3 groups of domains. They have different names within these groups. But basically what this is doing is collecting different annotations of the same domain. So, for example, here we see a domain that has one entry that starts with a CD here. That is the BLAST conserved domain database. But there's another one that starts with PFAM. So PFAM is a hidden Markov model-based domain database that happens to have its own model of the same domain. There is SMART, that's another one of these domain databases. Basically, it's collecting these and recognizing that although different databases may have slightly different predictions, they're all more or less picking up the same 3 domains here. So we can observe that we have these 3 different regions here, and we can also get some information about what's going on in each of these regions. So, for example, I can pick this NR LBD GR domain that we can see is the C-terminal region of the protein. We can get some information on this, and we can learn about what that particular domain does. Right, so that is going to bring up an entry on this particular domain. We can see that there's kind of a structural model, an image of what this domain looks like here. And we can get various information about what this domain does, as well as potentially useful things if we're trying to study this domain. For example, we can find out about structures that are related to this domain, so structures that are either structures of this domain or proteins related to it. That's often something we would be particularly interested in. And we can get information about how this domain is used in various other proteins that can help us form hypotheses about the function of this domain in our protein and in general. So, for example, this should be coming up shortly. It will show us models of the domain we were searching on, this NR-LBD domain, and various other domains found in different proteins that are associated with it. So each of these is a domain architecture, and this domain architecture is telling us, for example, we scroll down a little, that this NR-LBD domain that we were looking at, which basically seems to be some kind of hormone binding domain, is often associated with this middle domain here that we had seen earlier in the protein, and that turns out to be a DNA binding domain. So we can then suggest from this that usually this hormone binding domain, which forms this nice binding pocket for a small molecule ligand, is associated with DNA binding domains, and maybe get the hypothesis that this is some kind of hormone-regulated transcription factor. So it's something that maybe regulates DNA transcription and therefore gene activity based on presence or absence of hormones. We can see that in various proteins this is paired with various other domains. We have an estrogen receptor domain here, a glucocorticoid receptor. I think one of these is a retinoic acid receptor domain. Various other kinds of what are called modulator regions, which may suggest how this domain evolved and some of the versatility of its function. So this basic binding pocket is paired with a transcription factor domain, and that is paired with some kind of modulator domain that's affecting how it functions in different copies of this gene, so different homologs or orthologs of the gene, to maybe interact with different binding partners and thus regulate different functions. So any questions about any of that? Okay, so that basically is showing some of this domain information. But we can then go back to our last search here, and just a couple of things I wanted to illustrate in our blast results against the PDB database. So one of the things we can observe here is, in this diagram, we have our query sequence, and we've got essentially alignments to two different regions. We've got a bunch of alignments here to what we can now recognize is annotated as the DNA binding domain, and these are middle quality alignments. We don't have a structure that's a really good quality alignment here. And then we have good quality alignments to the C-terminal domain that we know is this binding pocket. We could scroll down to get a bit more information about them, but basically this is telling us that the modulator domain we can recognize as a distinct domain that varies quite a bit in this protein. We don't have a good structure model for that domain because nothing showed up in this PDB search. We've got relatively distant homologs, probably things that wouldn't be very good for homology modeling, to the DNA binding domain. And then we've got some really close homologs that are associated with the C-terminal ligand binding domain. And we can see the results of all of these hits here. We've got very significant hits, so E-values that are basically coming up to 0, very high identity. So probably, if we were trying to do homology modeling of this protein, we could do a good job with the C-terminal domain, probably not a very good job with the central domain, and we wouldn't be able to do a homology modeling of the N-terminal domain. Any questions about that? All right, so one thing we can notice here are these accessions. So accessions would always be links into a database where we can learn more about this particular hit to our blast search. In this case, you should learn to recognize accessions that look like this. So a number followed by 3 letters or numbers, and very often underscore and a letter. That particular pattern refers to a PDB ID. So an ID into the protein data bank, which I mentioned before is the standard repository of protein structures. If you remember these labels here, that would tell you what you would need to actually find an entry devoted to the 3D structure that produced this alignment here. So we could remember 1NHZ. And we'll say 1NHZ would be the PDB entry, so that would be one crystallographic structure in this case, or one NMR structure. The underscore A is referring to a particular polypeptide chain. So often a structure will have multiple polypeptide chains in it. Underscore A is saying chain A of entry 1NHZ in the PDB database. So just recognize those when you see them. Often that's what we really want if we want to get at the true structure of the protein. Any questions about that? So one more database that we've seen before that I want to go into because it has some nice structure information is Uniprot. So Uniprot, you may remember from the first week of class, contains Swissprot and Tremble, so these nicely annotated databases on protein sequences specifically. It also has some very good annotation on protein structure. So let me pick a term here. I'll type in glucocorticoid receptor. In this case, I'll throw in modifications. I'll make it an advanced search and say we want a particular organism, and I'll type in the organism ratus, so rat. So we're looking for rat glucocorticoid receptors. That should turn up some hits, and then we can look at what a Swissprot entry would tell us about this. So we've got an entry here. The gold starred thing is telling us it's Swissprot, so it's a human curated entry. It's likely to have a lot of good information. And if we scroll down, we can see some basic information on regions of the protein, but a lot of annotation. Some of this we'll see later in the term, but some of the things I particularly want to get to are the information related to structures here. So if we keep scrolling down, we can eventually get to, there it is, structure. And we can find some information on structures known for this protein. This will tend to have a good set of information. There are a few different databases we can use. Usually I would use RCSB, PDB, but basically it doesn't really matter too much. RCSB is kind of it's the US-based version of the PDB. That's kind of the original one. And we can learn about all the different structures that are associated with this particular protein. So we can see those IDs I mentioned before, one GDC, one GLU, one LAT, and we can see that there are a collection of different methods. Some of these are NMR structures, some of these are X-ray structures. And if we scroll down a bit more, we can see there's actually a fairly large number of structures associated with this protein. We can get some information in this third column on the quality of the structures, at least for the X-ray structures. So can anyone tell me, out of all of the structures here, which would be the best resolution X-ray structure? Well, remember that the quality of an X-ray structure is usually measured in angstroms to measure a distance. The smaller that distance is, the more precisely we pin down the locations of the atoms. So a good quality structure would correspond to a small number of angstroms resolution. The best one we can find here is 1.61 angstroms. It's a very good quality structure. So that can tell us we're likely to be able to get good models of this protein. One thing we do have to be careful of, just as we saw in our last search, is that, as I mentioned when we talked about X-ray solution, very often you can't solve an X-ray structure for a full-length protein. Often that's done at the level of domains. So maybe the whole protein is too flexible or to really crystallize nicely, but individual domains are more rigid. You can get a nice crystal. And we can see that that is the case with this protein. Basically, every structure of this protein that's available in the databases is a structure more or less the same region. Basically, the C-terminal region of the protein that would correspond to that ligand binding domain that we were studying before. So we have lots of structures of that domain. We don't have structures of other parts of the protein. And often that's something we would need to pay attention to. Any questions about this? All right, so we do also get some nice annotation in this database on protein domains. So we can see some annotation of particular domains within the region, that modulating domain. We've got the steroid binding domain, what they're calling the hinge, which is approximately where the DNA binding domain is. And we can also get information about various domain databases that have information on domains found in this protein. Just like with the CDD search, the Conserved domain database, you can see that there are different databases linked to this, so different structure or domain libraries that give us information on domains annotated to be in this protein. And some of these will pick up more or less the same domain. Sometimes one will catch a domain, another doesn't. Interpro tends to be a nice one. I can pick the graphical view. It's nice because it's a metadatabase that collects information from other databases. So like the BLAST CDD one, it will tend to give you kind of collected information that will catch what any one domain library misses. So we can see, for example, the glucocorticoid receptor. We have two different predictions of this domain. One of these, if we mouse over it, we can see comes from a database called PRINCE. PRINCE is a motif-based domain library, and the other one comes from PFAM. So PFAM is an HMM-based library. They're using different methods, but they both pick up the same domain in the same region. And here, just like with the BLAST one, we can see lots of different annotations, but basically 3 distinct domains found in this protein. As we saw with the CDD, you can click on any of these and get lots of useful information about these domains. We can learn that, well, there's some basic information known about the function of this protein. We can get information on architectures that use it. We can get information on structures known for that contain this domain, and so forth. So basically the same information we had seen before, more or less as in the BLAST conserved domain database. All right, so all of that is more or less showing you ways we can use resources we've already accessed to get additional information on structures and figure out when there are structures available for our particular sequences. But what I really want to get into, and I'll spend the rest of the lecture on, is the actual structures themselves. And usually when we're talking about structures, what we want to do is go to this database called Protein Data Bank, or PDB. That's one we haven't seen before. I'll just show you the link here. So we have a URL for that. It's pretty easy to remember, rcsb.org. If you just type that in, it'll work. But rcsb.org/PDB also works. And this is really the database of structures. It contains the structures themselves, and it contains a lot of information that's useful for accessing those structures. What I'm going to do is type in one of those PDB IDs we've seen before, 1N83, let it search, there 1N83, let it search for that, and that will pick up one entry into the PDB. So this is an entry devoted to a particular crystal structure that was solved for a particular protein, in this case, a piece of a protein, really, of a version of this protein we've been looking at so far. We can see a nice picture of what that particular protein's crystal structure looked like in a cartoon view. We can blow that up a little. We can see that this is mostly an alpha helical protein. So we have these coils that represent the alpha helices. And we can see the structure is solved with a ligand here, a small molecule. And if you read through the entry, you would see that this is a cholesterol molecule. Cholesterol may be the natural ligand of this protein. It's possible that it's not actually the ligand, it's just something that happens to bind nicely and lead to good structures, but at least in this case, that is found in this structure. We can get various other information that may be useful to us in studying the structure. So we can see here that this is an X-ray structure. What is the quality of this structure? Anyone see that? Is this a good quality structure? Yeah, so this is a 1.63 angstrom structure. That's very nice resolution. So this is a good quality model structure. So if we were trying to do structural studies with this protein, this would be a good structure to work with. It's likely that we've got a good model of this particular piece of protein. But we do have to be careful about the fact that this is very likely not the entire protein. And here we can see, if we look under this molecular description, that this is specifically residues 304 to 556 of this protein. So basically this is getting more or less that N-term or C-terminal domain, maybe some of that middle region, but it's not getting the N-terminal part of this protein. So that modulating region that doesn't seem to have a solid structure. We really want to go a bit farther than just looking at static pictures of it. We want to do some real analysis of what we can see in the three-dimensional structure. This is something I can only illustrate so much presenting to you here, but in recitation you're going to get a chance to work with these viewers yourself and see a lot more detail. But I will just open up this 3D view here. This is a Java applet, so I will say the possibility they've changed the security procedures here and this isn't going to work, but we will try to get this working here. If this doesn't work, I'll go back to the slides and I'll just say that you guys should play with this in your in recitation, see how to use these things yourself. But hopefully this will come up shortly. Yeah, so what this is giving us is a very simple Java-based viewer for molecular structures. If you really worked in a structure lab, you would want to use a much more sophisticated structure viewer. But here we see a basic viewer for molecular structures. And even this basic one has quite a lot of power behind it, a lot of different versatility, the things we can do. And I'll ask you in recitation, just play around with these, try some of the options I'm not showing you today, but I will at least try to give you a basic idea for what we can do. So of course, we can move this around, zoom in and out, try to get an idea of how this is working. And if we were trying to study maybe the enzymatic function of this, how it interacts with its ligand, we might be interested in maybe zooming in on the binding pocket there, so trying to get a bit closer view of what's going on with that ligand. And we may want to play around with how we're viewing this particular protein. So if we right-click here, we can pick a style and see that maybe we pick the scheme of space filling. That might give us a somewhat better view of the detailed interactions going on in the region of that ligand. Now in this case, there's not a lot we can see here because this has gotten kind of messy now that we are space filling this. But we have a lot of flexibility in these things if you really get into the details of how these work. So I'm going to open something called a console here that lets us do a lot of more sophisticated things than you can just the mouse. And what I'm going to do is type some commands here. So select all, color gray, turn the protein gray. I'm going to type select hetero. Hetero refers to things other than the protein. I'm going to type color red. And that's going to bring up everything that isn't protein highlighted in red here. So you can see the ligand. But we've also got a lot of these oxygens that seem to be just sort of sitting around doing, not bound with the protein. Can anyone guess why there are oxygens sitting around the protein? Those are water molecules. So very often when you work with a structure, you need to model the water around it. You don't generally show hydrogens in these structures, so water molecules just show up as oxygens. So what I'm going to do is select water. I'm going to turn the space fill off so the water goes away. And now we've got a much nicer model of the binding pocket where that ligand occurs. And if we were trying to study enzymatic function, we could look at this and get a pretty good idea exactly which atoms might be interacting with that ligand and key to the behavior of it. So if we wanted to know how we could maybe mutate the protein to make that binding stronger or weaker, then we could get good hypotheses by looking at exactly what's interacting with this kind of structure. There's a lot more we can do with this, a lot more versatility that I'm not going into here, but that gives you some basic things we can do. There is one other thing I want to illustrate because I want you guys to try this in recitation. So let me just try select all, turn off the space fill, turn off wireframe, which is these the connections of the bonds here, turn on cartoons, so that's bringing back the cartoon view of the protein. And one of the things that I think is just really neat to do is go to this style, look for stereographic, and pick cross-eyed viewing. So what this is showing, you see it looks like you've got two different copies of the protein side by side. So cross-eyed viewing is a way of viewing 3D information on a 2D surface. And this is something that, if you've never seen it before, it's just, I think, a neat thing to see, and I'll ask you to try this in recitation. So it takes a little practice to be able to use these, but once you've done it a few times, you can just get it immediately anytime you see this. This looks like two copies of the same protein, but they're actually slightly different. And the reason they're slightly different is that you try to use them in a way where each one is essentially contributing to one of your two eyes, and your brain basically can put that together to make a model of the three-dimensional structure. And the way you use this is, you look at it and you kind of cross your eyes so that you're seeing double. And then you try to make the right copy of the left protein sit exactly on top of the left copy of the right protein. And if you get it just right, then it'll snap into focus and you can see the full three-dimensional structure of the proteins. Once you've got that, you can try spinning it around, moving. Basically, you see 3D on the 2D screen. I will say that this is not something you want to spend a lot of time doing. It gives you, the eye strain from this is pretty bad. If you really work in a structure lab, you would use 3D glasses to see these kinds of things, but, or specialized goggles. But this is something that you can try on your own. And if you're doing a poster, you want a 3D picture in a scientific paper, this is something you can use to convey 3D information to anyone who wants it. So if you haven't seen this before, it's very unlikely you can get it from the screen. You really want the position very nice, but that's something you can do with this. And again, I encourage you to spend a few minutes in recitation and try to get this working for yourself. So is anyone able to see this right from here on the screen? Okay, so some of you can get it working. It'll be easier in recitation when you've got the computer right in front of you and you can position it optimally. Okay, so there's one last thing I wanted to talk about before we break for the day, and that is searching with structures. So you should have heard a bit about that on Wednesday. I want to show you a couple of ways we can do searches of types we haven't seen before. So we've seen before how to search for the keyword, how to search for the sequence. You also have some basic ability to search with structures. So if we go back a page here, and we pick the summary page, that's our starting page, we can see there are a couple of different ways of searching with structures. There's an automated version of this where you can click 3D similarity. You click a comparison method here, so I will pick JFATCAT flexible. And this is a structure-based search. And what this is going to do is run another Java applet to visualize this. And what it's done is search the structure database for similar structures to our current protein. And it's overlaying them in two different colors here. And we should be able to see we've got a really nice model here. So there is another structure that is very close to the structure we're looking for. So that is not quite as mature a technology as sequence searching, but it is doable. You can now do these kinds of searches and find structural homologs to your proteins. Very often, if we really want to do this, what we would want to do is look at pre-computed hierarchies of structures. Let me just go back here. Go back to the main page. And we'll see at the bottom of this page, there are a couple of links under external domain annotations to databases where people put together big libraries of related structures. So one of the popular ones is called SCOP. There's now some more advanced versions, SCOP-E and SCOP-2, that are a bit more sophisticated than this. But SCOP is a nice one that, if we click on this, shows us that we have our particular protein here, and then that is classified as an example of a domain, which is classified into a family of domains, which is classified into a superfamily of domain families, which is classified into a general class of folds. And if we wanted to find other homologs of this protein, we could pick, let's say, the family in which this domain is found. It will tell us all of the PDB structures related to that. We could narrow it down by saying that maybe we want an NMR structure. and we could get to an NMR version of this particular structure. We can pull up an image of that NMR version, and we can see that in this NMR version, we've got some dynamical information. You can see lots of little chains here. What this is doing is overlaying different predicted structures for this that came out of the NMR. So we can see an overall structure and also get some information about maybe flexible versus less flexible regions of the protein. Any questions about that? So one last little thing I did want to demonstrate here that is useful when sometimes when working with the PDB is for those of you who can program, sometimes you really want to get into the meat behind this, actually work with the structures. If you download the PDB text file, I'll just click a browser, we can use WordPad will work here, open it with WordPad. You can see what's actually behind a PDB structure. So there's a text version of the structure, has lots of annotation related to it, but also detailed information on every atom in the structure. So we can see that there is a carbon atom that's the alpha carbon of alanine 209. It's at a particular position in 3D space in this structure. If you were actually writing computer programs to work with structures, this is what you would work with. You would want to download this, relatively easy to parse, and you can get a detailed 3D model that you can work with your own compute code. So I think we are out of time here, but I again encourage you to play around with some of these things in recitation. Are there any last questions before we break for the day? Okay, if anyone has homework, please make sure I have these here.